Any questions about DNA replication? A summary of DNA replication then. You just follow this, counting 1, 2 to number 5. Helicase, these purple blobs here at the replication fork are opening up the replication fork and straightening out the helix. Single-stranded binding proteins help to stabilize that single strand of DNA until the DNA polymerase gets into place. The leading strand, because of the orientation of the leading strand, the DNA synthesis of the complement of the leading strand can proceed in the same direction as that replication fork is opening up. But because this strand is going in the opposite direction, DNA replication needs to go in this direction, not in the same direction as the replication fork is opening up. So you can see back here, this is where the replication fork used to be, right? The opening of the replication fork used to be back here, so the primer was put into place here by the primase, and DNA replication happened in this direction. But while it was happening in this direction, the replication fork opened up more behind it. So we have this primer up here being put into place. Okay, that's where the replication fork used to be opening up. And DNA replication happened in this direction again. But then the replication fork opened up more behind it. So we need to start DNA replication again behind that. And the consequence of this discontinuous DNA replication is that you get these small fragments of DNA being synthesized. And those fragments are called Okazaki fragments. Now, what you see right here, here's the DNA polymerase 3 synthesizing this piece of DNA. Okay, let's go back up to this one. This DNA polymerase 3 is going to encounter that primer. Okay? So once it gets close up to it, that DNA polymerase 3 is going to be substituted for DNA polymerase 1. And DNA polymerase 1 is going to chew through that. It's going to take the nucleotides of the RNA primer off and replace them with DNA nucleotides. Okay? And then finally, there's going to be a little gap like this, and that gap is going to be filled by ligase. It's going to ligate those two DNA fragments together, as is happening right here. We also talked about proofreading. So the process of DNA replication is very high fidelity. You know, it doesn't make very many mistakes at all. But we need it to be even better than it is. So we have some mechanism for recognizing mistakes. Some of that mechanism is done by the DNA polymerase itself. So as it's synthesizing DNA, it's able to feel topographical indications or bumps that indicate that the wrong nucleotide was put into place. Okay? But sometimes so that's the DNA proofreading mechanism of the DNA polymerase. We also have other enzymes that scoop down the DNA after the DNA polymerase has done its job, and they can recognize individual nucleotides that are mismatched, okay? And they can just flick out those nucleotides. They can excise individual mismatches. So they take out that nucleotide and replace it with the proper nucleotide, one that complements on the complementary strand. Another way of approaching it is a different enzyme altogether can recognize that this nucleotide is incorrect, it doesn't fit into this spot. So instead of removing that single nucleotide, it'll remove 5 or 6 of them and then fill in that whole gap. Okay, so that's excision repair. It's excising a small sort of tetranucleotide, pentanucleotide, and then filling in that whole gap. Any questions about DNA replication or proofreading or any of this? Okay, let's shift gears now. This is how we're going to start talking about how DNA functions as a recipe for the proteins in our body. Something called the central dogma of molecular biology talks about the direction or the directionality of information from the DNA to the RNA to the protein. And this is a one-way street. Okay? We get information from that core information source, the DNA, in the genome, in the nucleus, if it's a eukaryote. That can be transcribed into an RNA copy. Okay, so that's what's happening up here. That transcription process is enabled by an enzyme called RNA polymerase. Okay. This transcript, or mRNA, messenger RNA, will be exported out of the nucleus. So here we have an indication of the nuclear membrane. That messenger RNA then gets translated into a protein that happens in the cytoplasm outside the nucleus when we're talking about a eukaryote. Okay, so these are the two main processes then, so that we can get information from the nuclear genome, or just the genome in prokaryotes, transcribe it into a copy that can then be translated into a protein. So what we've got here is the flow of information from DNA to RNA to a polypeptide. The first step of that is transcription. So the enzyme RNA polymerase sits down at the beginning of a gene, opens up. It looks kind of like a replication fork, okay? The difference is that the mRNA that's being synthesized, okay, just like a DNA molecule would be synthesized, doesn't stay base paired to the template for that mRNA. Okay, what happens is that the DNA comes back together. So this mRNA copy then becomes a single-stranded molecule. Okay, so it's bound. The synthesis of it happens just the way the synthesis of DNA happens. You get complementary RNA nucleotides, base pairing to the DNA. They get polymerized, okay, and the process of forming that phosphodiester bond is the same process, okay, but they don't remain base paired. The DNA comes back together and forms that double helix. So the replication bubble then is a transient thing. Okay. Now, you've seen lots of differences between eukaryotes and prokaryotes. You know, eukaryotes have membrane-bound organelle, prokaryotes do not. We've talked about how metabolism is different. Glycolysis happens in the cytoplasm, and the Krebs cycle happens in the mitochondria. Obviously, the Krebs cycle can't happen in the mitochondria of the prokaryote, because they don't have it. Another big difference between prokaryotes and eukaryotes is the structure of genes. Okay? In prokaryotes, a gene is transcribed, and a transcript is directly translated. In eukaryotes, there is more complexity to the gene. Okay? The gene has alternating sections of what we call introns and exons. Okay? Introns are intervening elements. These are non-coding sections of the DNA. Okay? So when a transcript is made in a eukaryote, that transcript we call a pre-mRNA. Okay? And the pre-mRNA needs to be edited, okay? Because it contains all of these intervening elements. Okay, so here we have an exon in blue and an intron in yellow. So the only coding part of this gene are the blue sections. Okay? So when we make a transcript from it, we have to edit out the copies of these yellow sections. And the amount of the yellow sections, the amount of introns in the gene can be huge. Okay? Here's just some examples. The globin gene, 1,500 base pairs long. More than half of it, 900 bases of that are introns. Okay? In the albumin gene, 7,500 base pairs long. The exons are only 1,800 bases of that. Okay? Here's another gene, 10,000 bases long. The exons are only 2,200 bases of that. So there's 5 times as much sequence that are in introns as are coding. So here's an electron micrograph of a newly transcribed mRNA on the top. And what you've got is that mRNA is base paired to the pre-mRNA is base paired to an mRNA. So what we have here is base pairing with all these loops, and all these loops are the introns. Okay, and all those loops need to be edited out. Okay. So here is a processed mRNA base pairing to the gene. Okay, so you can see all these introns looping out where the introns have been excised, they've been edited out of the mRNA. So let's just talk about expression for a minute. We'll come back to the structure of an mRNA. So we know that information flows from the gene to the mRNA to the protein. We know that that's the sort of interface between the genotype and the phenotype. Okay, this process of transcription and translation takes us from the genotype to the phenotype. And we know that we have a whole lot of cells in our body. And all the cells in our body are doing different things. And they're not expressing all of the genes all of the time. So if we just look at some examples of that, if we look at a pancreatic cell and the genes that are expressed in a pancreatic cell versus a white blood cell versus a nerve cell, if we just look at 3 or 4 genes, we see, of course, glycolytic enzymes are going to be expressed in all cells. Okay, all cells of our body need to metabolize, okay, with the exception of mature red blood cells. Pancreatic cells, of course, need to express the insulin gene, but the insulin gene doesn't do a white blood cell any good. Okay, so we don't express pancreatic or insulin genes in white blood cells, nor do we in nerve cells. Okay? White blood cells, however, we express a whole suite of antibody genes, but they don't do nerve cells or pancreatic cells any good. Okay? So how do cells know? How do we make sure that cells aren't wasting all their time expressing genes that they don't need? Okay? We don't have anywhere near those kind of resources, neither in terms of the amino acids that would be necessary to make the proteins, nor the ATP that would be necessary to drive the chip. So we can look at a very simple model in bacteria. Bacteria, obviously, by extrapolation, can't express all of its genes all of the time either. It doesn't make any sense to do that. So there's a long history of selective pressure enabling bacteria to express only those genes at the time that they need to express those genes. And this is actually a very, very big field right now. As we've gone through and sequenced entire genomes of organisms, that's one level of information, understanding the genome of an organism, but understanding how that genome is expressed, both where and when. Okay? This is what we call the transcriptome, or the proteome. Taking a look at when things are expressed and where they're expressed. Okay, that makes a huge difference to what kind of cell something is and how it functions, both in space and in time. So just a little bit of background. We often see genes organized in something called operons. So operons are clusters of genes that share, that have similar function, or they have a related function.